Alright, today I'm gonna talk about recombinant DNA. So, first off, let's begin. What is recombinant DNA? Recombinant DNA molecules are DNA molecules formed by laboratory methods of genetic recombination that bring together genetic material from multiple sources, creating sequences that would not otherwise be found naturally. So what that means is that it's something created by scientists in laboratories. So it's a different DNA that's created by by scientists in laboratories, and how that's made by many steps. So let's see them. Okay. First step is isolation of genetic material. The first step in in recombinant DNA technology is to isolate the desired DNA in its purest form. That means free from other macromolecules. Then we go into step number 2, restriction enzyme digestion. Restriction enzymes act as molecular scissors that cut DNA at specific locations. These reactions are called restriction enzymes digestions. Then to step number 3, amplification using PCR. PCR is a method of making multiple copies of a DNA sequence using the enzyme DNA polymerase in vitro. Step number 4, ligation of DNA molecules. The purified DNA and the vector of interest are cut with the same restriction enzyme. This gives us the cut fragment of DNA and the cut vector that is now open. Step number 5, insertion of recombinant DNA into host. In this step, the recombinant DNA is introduced into a recipient host cell, mostly a bacterial cell. This process is called transformation. Step number 6, isolation of recombinant cells. The transformation generates a mixed population of transformed and non-transformed host cells, and that's how to make a recombinant DNA. So this is just like a summary of it. You can pause the video if you want to look at it, but this is just a summary of how recombinant DNA is made and an example of it. So over to the uses of recombinant DNA. Recombinant DNA is widely used in biotechnology, such as and medicine and research. The most common application of recombinant DNA is in basic research, in which technology is important to most current work in the biological and biomedical sciences. Many additional practical application of recombinant DNA are found in industry, food production, human and veterinary medicine, agriculture, and bioengineering. Through recombinant DNA techniques, bacteria have been created that are capable of synthesizing human insulin, human growth hormone, alpha interferon, and other medically useful substances. So what that means is that recombinant DNA is, is something that's really good for humans. If recombinant DNA is used correctly, it can be used in making many stuff, such as medicine, changing food, uh, making different fruits, and so much more. These are just the simplest stuff. That's all. Thank you for watching.